Hello and welcome to the Institute for Molecular Science and Engineering's webinar, Cradle-to-Cradle Engineering for Polyolefins Tailored for Biodegradation. This webinar will be presented by Dr. Alice Banks from the Department of Life Sciences at Imperial College London. My name is Leah Adamson and I'm the Events and Communications Manager for the Institute for Molecular Science and Engineering, or IMSE. So this webinar is part of the IMSE Seed Funding 2022 Projects to Solve Today's Grand Challenges series. Each webinar in this series highlights a project that was funded in this round of seed funding held by the Institute for Molecular Science and Engineering. All of these projects use molecular scale science to solve societal and industrial challenges. The theme of this year's competition was sustainability and circular economy. So before the webinar starts, we're going to be putting some helpful links into the chat function, which you should be able to see now. So these links include the IMSE Eventbrite page, where you can find the registration for all of our upcoming webinars in this series and beyond. The IMSE YouTube channel, where you can see all our previous webinars, and this webinar will be uploaded. Our IMSE blog, the Never Lick the Spoon podcast, all of our previous briefing papers, and registration to our IMSE newsletter so you can keep up to date with all of our news and events. I'm also going to put the link to a news article where you can find in the information about all of our 2022 IMSE seed funding projects. So just a few top tips before we start the webinar. If you wish to ask a question, please put the question into the Q&A function at any time during the webinar. Your questions will be answered at the end. Please do not put questions pertaining to the presentation into the chat function. If you have any technical difficulties, please message IMSE Imperial privately in the chat function, and an IMSE team member will try and help you where possible. So I'm going to now hand over and introduce our presenter for today. Our presenter is Dr. Alice Banks, and they're a postdoctoral research associate in synthetic biology at Imperial College London. She's currently working as part of the ERA Chirology Tech Consortium, microbial integration of plastics in circular economy. Her research focuses on engineering bacteria and microbial communities to degrade plastic waste and upcycle degradation projects into new value-added materials. Prior to her current role, she worked at Newcastle University, developing biologically responsive materials using cell-free protein synthesis systems. This followed a PhD at the University of Bristol in the field of fungal genetics and molecular biology. Alice is also a dedicated STEM ambassador and a member of the iGEM community. She has experience working as part of outreach, public engagement, and widening participation initiatives. So I'll now hand over to Alice and we'll hear her presentation. Thank you.
Thank you very much, Leah. And thank you very much for the opportunity for me to share this work as part of the IMSE webinar series. I'm really pleased to be here, and it's exciting to be kicking off the whole series with the first talk. So the project that I'll be talking about today is cradle-to-cradle engineering of polyolefins for tailored biodegradation. And this is a short project which forms part of the seed funding programme and just began at the start of the month, so we're still in fairly early stages of this work at the moment. So as I'm sure you're aware, plastics are really ubiquitous in our everyday lives. And although we probably think of plastics as a fairly modern material, there are actually examples that date back to the 1800s. And the first of these is Parkesine, which is generally considered to be the first of the manufactured plastics. And it's a celluloid material, and it was produced using cellulose and nitric acid. And initially, this was developed as a cheaper and a more colourful alternative for items that would typically be made from tortoiseshell. So this would include things like combs, brooches, hair clips, and sort of beauty artifacts like that. And many of these pictured here are actually just on display at the Science Museum. So for anyone at the Imperial South Kensington campus, that's just next door. And then moving on towards the end of the 1800s, we then have the production of galalith, which was produced using casein and formaldehyde to harden it. And this was used for some similar items, but particularly in the production of buttons and other plastic type materials that would form parts of garments, as it was fairly durable and therefore able to withstand the regular washing and heating processes that you encounter in the laundering and ironing. So these are two quite early plastics. However, it was then really only from the 1900s when plastic manufacturing really took off, starting with Bakelite, which was used in the production of all sorts of domestic items and appliances. And Bakelite became the first plastic that was then derived from fossil fuels. And the production of this material then sparked the research and development of a whole wealth of oil-derived plastics in the first half of the 20th century. And many of these are now very, very commonplace in our everyday lives. And you probably encounter a lot of these things every day. So we have materials such as cellophane, which can be produced thin, transparent, so is therefore useful for sort of films and packaging. Polylactic acid, or known as PLA, which is what many of your compostable drinking cups and things are made from. Polyvinyl chloride, or PVC, used for things like car seat coverings, so fairly tough. Polyethylene, there are different kinds, but this is used for lots of packaging products, whereas nylon is a material quite commonly used in garment and fashion industry. We then have other materials such as polystyrene, used in packaging again, and polyethylene terephthalate, or PET, which is the material comprising most drinking bottles and many food packaging products. And then also polypropylene, expanded polystyrene, used as an insulation material in the building industry. And another example is acrylic, developed somewhat more recently, which again has good transparency, so is a really useful material. And the use of these globally has just boomed. They're used in all aspects of our everyday lives because they're so versatile and such useful materials. However, it's only really been in the last 30 years that interest has grown in bioplastics, which has been spurred on by the impact of plastic pollution and climate change, which we've been acutely aware of more recently. So although we've ultimately reached the point where we cannot live without plastics in our daily lives, there's now the really pressing need to develop more sustainable options and alternatives to use instead of these materials that are derived directly from fossil fuels. So here are some statistics which really sort of prove this point. We have reached the point where we're at a plastic crisis, where plastics are used and disposed of globally and on a vast scale. And these statistics presented here are taken from data that's been compiled by the UN. And it's really clear to see the really devastating impact that plastic use is having globally. So apart from the obvious drain on finite resources, you can see here 17 million barrels of oil are used annually for plastic production. There are also many environmental impacts that are resulting from the poor and insufficient disposal and recycling practices that are currently in operation for plastics. And escaped plastics are finding their way into pretty much every corner of the world. They're polluting the lands and the oceans and causing irreversible damage to our ecosystems. It's particularly true of plastics which enter the oceans, because once they're in the oceans, they're really quite irretrievable. And waste plastics are having an impact, therefore, those that are in oceans, things like marine animals and plastic that's contaminating the oceans. But waste plastics are also having an impact on human health. And we're now understood to be consuming plastic particles in a huge portion of the water we drink and the food that we consume. You can see here that 90% of bottled water was found to contain plastic particles, and 83% of tap water was found to have the same, have plastic particles as well. And quite worryingly, there's been some recent papers that were highlighted in the media in recent months that have also reported evidence of plastics appearing in human blood samples and also in lung tissues. So this is really quite worrying that it seems so inescapable. And there's therefore a particular need to address plastics which escape the intended refuse and recycling streams, as these are some of the ones that are causing most impact in terms of plastic pollution. So I'd like to speak a little bit more about fugitive plastics, and these are the escaped plastics which result from littering and leaked waste that ends up being left out of the refuse collection system. So looking to the right hand side here, this is some data from a few years ago now, 2015, that was put together by the Ellen MacArthur Foundation. But if we consider just the plastic waste which results from packaging on a global scale, then about 86% of packaging products end up being disposed of and entering either the waste or recycling routes. But of that, about 40% goes directly into landfill, so that without even an attempt to recycle this. About 14% is collected, so it's more likely to go down the intended recycling routes. We then have a similar proportion of which just ends up being incinerated, but about a third of all plastic packaging products ends up escaping their waste or recycling route and becoming fugitive plastics. And they therefore avoid any downstream processing routes. And this can be the result of deliberate littering, which we're probably all aware of and probably again see all too frequently. However, it can also be the result of waste being transported or blown away from where it had originally been disposed of. But it is expected that by 2050, there'll be an estimated 2.1 billion tonnes of fugitive plastic waste globally. And unfortunately, around 80% of this fugitive plastic ends up making its way to the ocean. And as you said, once this waste enters the ocean, it's incredibly difficult to retrieve and process. So the reason a lot of this actually reaches the ocean is through sort of blowing around and entering rivers and waterways that ultimately leads to the sea and the oceans. It's not necessarily that these plastics have been dumped into the sea intentionally. However, this is also the case in some areas as well. And when we look at the composition of these plastics, then a huge proportion of plastic packaging is made from plastics which fall into the material class of polyolefins. And it's this particular class of materials that are the focus of this IMSE project. So polyolefins are polymers, and they have this general formula shown here in bold, where the R group is an alkyl group. And there are some examples here, both the generic format, but also the R groups which either make polyethylene or polypropylene. And these R groups can vary quite considerably. And this then gives rise to a whole group of polymers with a really diverse range of physical properties. So we can have variation in the molecular weight and the crystallinity. And this then goes on to affect the rigidity and the properties of this polymers, which creates a hugely versatile group of materials. So we have things with very low crystallinity, such as adhesives, which are basically liquid-like with incredibly low or no crystallinity at all. And right up at the other end of the spectrum, we have materials with a really high proportion, where over half the material has crystalline properties. And these are the much more rigid plastics, which can be suitable for more structural building materials or things where structural integrity is really required. But the versatility and the relatively low cost of producing these materials means that they're really widely used in everyday products. So within polyolefins, some of the most common classes are polyethylene and polypropylene. And polyethylene can be produced as two main types, either low density polyethylene or high density polyethylene. And low density polyethylene is softer and it's more flexible. And it can have quite a high degree of transparency as well. So that makes it suitable for materials such as films where transparency is desirable, but also softer plastics such as bags. So a lot of your standard supermarket carry bags are going to be made from low density polyethylene. And high density polyethylene is far more rigid, and it also offers a greater level of chemical resistance. So this means it can be used for things like packaging cleaning products and other chemicals where they need to be stored and transported in a safe material. Then if we consider polypropylene, then there are some different properties. And what polypropylene is very good at is it has good resistance to fatigue, which means it can be used for items which undergo repeated actions, particular movings, sort of moving parts, such as the bottle cap shown here, that undergo a lot of twisting and stress. but also things like sports cap, bottle caps that flip open and closed and are expected to be opened and closed repeatedly, and they need to be able to do that without shearing under the stress. So 2 different types of polyolefins, both with different desirable properties for different intended purposes. And the challenge that comes with this is that because they have so many useful properties, they're very widely used, particularly in commodity packaging. And many of commodity packaging items tend to be single-use items, which are just intended to be used and thrown away. Now, within the UK, we have the UK Plastics Pact, and this sets out several key goals which should be, which we have a target of reaching by 2025. So in the first of these, there's initially a real drive to eliminate the use of any unnecessary or single-use packaging through the removal or replacement of it. And this should ultimately minimise the volume of plastic waste that is being disposed of at all. And additionally, by 2025, all packaging within the UK should be suitable to either be reused, recycled, or composted. So there should not be any packaging which is being sold, which is just intended to be used once and disposed of in for landfill. We then have the goal that 70% of plastic packaging should be effectively recycled or composted. So although 100% should be suitable for these routes, it's likely that some of it will simply be thrown away. So we hope that 70% of this will enter the intended routes. And finally, across all plastic packaging, there should be an average of 30% recycled content going into plastic packaging as it's produced. And this should help to reduce the demand for virgin materials, which are currently used for a lot of packaging products. So in the work that's in this IMSE project, then we'll be considering these goals as we're addressing the biodegradability of polyolefin materials. So the project that we're just starting out on is a collaboration across departments of life sciences, where myself and Jose work, and aeronautics, where Kenyang is based, both at Imperial. And we're also working with an external collaborator, Florence, who is based at a company called Polymateria, who have developed a polymer additive which enables the safe and complete degradation of plastics in our environment. So I'll just tell you a little bit more about the technology that Polymateria have developed. And they have a novel additive technology which is able to accelerate the biodegradation of plastics in the environment. And this additive technology is available to manufacturers in a pellet form, so it can be incorporated with the resins during the production of plastic-based packaging products. And it's designed such that after a specified dormancy period, this technology is activated, and this allows it to attack the crystalline and the amorphous regions of polymers, which converts them from long polymerized plastic chains into depolymerized waxes. And these much smaller waxes are then far more available to environmental bacteria and fungi that be encountered in the environment. And these microorganisms are able to utilize the waxes as a sole carbon source for food. Now, a huge benefit to this technology developed by Polymateria is that it operates at mesophilic temperatures, which are the type of temperatures that we encounter in the UK. Whereas if we compare this to some other biodegradable plastics, particularly things like compostable soft plastics, they often require elevated temperatures for successful and complete degradation, which are unlikely to encounter in a lot of parts of sort of Northern Europe, for example. So moving on to sort of the aims of our IMSE project, then we've sort of divided it into 3 main aims. And the first of these is to manufacture novel materials with bespoke designs. So this will include the incorporation of additives, such as the technology which has been developed by Polymateria, which is expected to aid the biodegradation of plastic products. And we'll also explore a whole range of polyolefin materials which have been produced with things like a different range of percentage crystallinities, and also some which have been exposed to weathering pre-treatment processes, which is things like wind and exposure to UV light. Secondly, we're going to be drawing on the capabilities of microorganisms and using these as a means to degrade these candidate polyolefins that have been developed in the first aim. So we'll be screening microbes and microbial communities for the ability to degrade polyethylene and polypropylene. And we'll be both looking for the naturally occurring ability for microbes to carry out these degradation activities, but also we're able to culture organisms in the presence of polyolefins, and we expect them to then adapt the ability, evolve the ability to degrade materials through these particular selection pressures. And in our final aim, we're hoping to gain a better understanding of some of the mechanisms which are at play in the biodegradation processes. And specifically, this means that, identifying intermediate products that are formed as part of degradation and elucidating the enzymatic pathways that are involved with that. So this work actually follows on from some preliminary investigations that were carried out by an MRes student who was working in the lab last summer, called Murin. And Murin focused on the use of microbes to degrade high density polyethylene, as in some research she found that the majority of polyethylene biodegradation research had really focused on low density polyethylene, and high density had been somewhat neglected. So in doing this, she explored a whole range of high density polyethylenes, which is listed in this table here. And she had samples with a range of different properties. And she isolated organisms from the soil and screened these microorganisms for the ability to degrade the panel of polyethylenes. So in her setup, then the inoculum came from a soil sample, and polyethylene then provided a sole carbon source, which meant that any microbial growth that was observed would be the result of utilization of these polymers. She then went on to identify the microbes, which were successful by sequencing a region of the genome called the 16S. And in a final examination, Murin went back and looked at the polymer powders that had been degraded by microorganisms and examined them for any changes resulting from degradation. So, I'm now going to present some of Murin's results, and she found that in her growth assays, 3 of the 5 polymer samples that she tested resulted in an increase in the optical density, and this was, for these 3 polymers, it was significant compared to the control samples. So these were polyethylene sample 1, shown in bright blue, which we can see here, whereas the control sample, there was no increase in optical density over the time tested. Also polyethylene 2, which is in here in black, and 5 shown here in green, which were fairly similar to one another. And again, their control tubes didn't show any growth. And just say the control tubes is when no inoculum was added, so there shouldn't be any microbes available. So 1, 2, and 5 all showed degradation when compared to their control and growth. If we look at these ones here, which is polyethylene sample 3, it's not quite so clear, because although we have seen an increase in optical density, this is the case in both the test and the control tube. So it's possible here that over the period of incubation, then the polymer changed in such a way that it increased the turbidity of the sample. So we can be less confident with this, but the other 3 here all looked as though the microbes were able to grow using the polyethylene that was available. So to corroborate the experiments from monitoring the growth, Murin also collected data on the abundance of microbial colony forming units at each of the different time points. And she found that these data agreed with the previous data set and seemed to make sense in that all of the control tubes showed no growth at all. So that's what the gaps are for each of the 3 time points here. And another thing that agreed was that the data seemed to plateau after about 2 days. So there was an initial increase in microbial growth in the first 48 hours of the experiment, but after this, there was very little change. Another advantage of carrying out this experiment, where she plated out onto agar plates the microbial cultures, allowed her to obtain individual isolates and pure samples, which could then go on to be identified using amplification of the 16S region and subsequent sequencing. So she was able to sequence a fair number of the isolates that were successful at growing on polyethylene by sequencing the 16S region. And this revealed that the majority of isolates that were successful at degrading polyethylene belonged to the Pseudomonas genus, which is a common soil bacterium. And some were also found to be belonging to the Acinetobacter genus. Now, the Pseudomonas species in particular often have the ability to form biofilms, so it's possible that this trait has helped them to colonize the plastic particles that were available in the experiments and therefore exploit the polymers as a sole carbon source. And as well as looking at the microbes themselves, Murin, in collaboration with Florence, based at Polymateria, went back and recovered the plastic particles, and these were examined using a process called gel permeation chromatography. And this allows us to examine the molecular weight and the relative size of the population of plastic particles remaining. So these were examined by looking at the number average and weight average molecular weights, which leads to this polydispersity index, which is the metric that we're particularly interested in. And this is a metric which indicates the heterogeneity of the sample. So a higher index suggests that there was a larger range of particle size, whereas a small number suggests that the particles were all fairly similar to one another. So I'm just going to point out a couple of examples in here. And if we look at polyethylene sample 1, this was one of the polymers that led to good microbial growth. Then the PDI index in the experimental sample is lower than in the control. So this may suggest that many of the particles available of one particular size have been consumed, and this has left some that are less accessible to the microbes. And another example would be polyethylene 2, and we can see that the inverse is true here. So in the test experimental sample, then we've got a higher PDI than in the control tube. And this might be the result of microbial activity cleaving many of the chains, leading to a whole range of different polymer sizes. Another reason could be that some of the particles may actually be larger due to the result of biofilms forming on these. I mentioned that Pseudomonas strains are quite prone to forming biofilms, so this may have affected the size of some of the plastic particles that are available. So this is fairly sort of preliminary data at this stage, but it's given us a bit of an indication of how the microbial communities are changing the properties and characteristics of these plastic particles. So a final set of experiments that Murin ran was, instead of looking at polymer powders, she was looking at plastic films. So she was using polyethylene films that actually came from carrier bags. And these hadn't undergone any kind of pre-treatment conditions, so they'd not been weathered or exposed to UV or ground up at all. They were just a piece of post-consumer plastic waste. And she looked to see if microbes from a range of different sources, so she used microbes that come from the soil, a standard E. coli strain as sort of negative control, and a microbial strain called Pseudomonas, which we have in our collection and is quite promising in terms of its activity and application in biotechnology. And finally, a control sample where no bacteria were included. And she tested the growth of these in the presence of polyethylene bag in a rich medium, so LB, and also in quite a minimal medium, which was M9. And after an incubation period, we then examined these films using scanning electron microscopy to look at any changes in the surface morphology of these polyethylene films. And what the SEM showed was that it was fairly clear that for bacteria that were isolated from the soil and also the bacterium Pseudomonas, we saw quite marked changes when the bacteria were grown in the rich substrate with the plastic present as well. And there was some holes, some corrosion, and quite significant change to the plastics in both of these instances that we didn't see in our negative control with E. coli, And we also didn't see where we just had the polyethylene in the liquid broth under incubation. So what this might suggest is that it's really important to include sources of microbial nutrition if we're hoping to degrade polymers using microbes, as it's likely that the presence of reasonable level of nutrition is going to help support the formation of a biofilm, which is ultimately going to lead to a better level of biodegradation in the environment. So to conclude sort of Murin's work, she found that soil-dwelling communities offer a really promising source of microbes to degrade polyethylene, and that soil microbes are able to liberate the plastic monomers and use these as a sole carbon source for growth. And one of the most abundant genera are Pseudomonas, and also Acinetobacter to an extent. And these isolates also seem to be active in degrading post-consumer pet films, particularly in the presence of a rich medium. And the data that Murin collected during her master's project has led on to this IMSE proposal, which we've recently started. So I'm going to next talk to you about what we're hoping to achieve here. So we have three main objectives to carry out, and the first of these is to produce a range of different polyolefin materials, both polyethylene and polypropylene, that will be manufactured with a range of different compositions and properties. So this is going to include materials manufactured to include recycled content in line with the UK Plastics Pact. Also materials exposed to harsh weathering and pre-treatment conditions, and then samples which contain this technology which has been developed by Polymateria to aid biodegradation. And then in our second objective, we'll be screening soil isolated microbes for their ability to degrade our panel of polyolefin materials. And finally, in our third objective, then we aim to elucidate the degradation mechanisms that are at play through chemical analysis, which should indicate the intermediate products that have been produced. So the experimental strategy that we're taking will initially focus on polyethylene, as this is one of the most abundant plastics and it's really favoured for its strong, flexible and heat resistant properties. So we have a range of different polyethylene samples shown here, which, as I say, include the technology developed by Polymateria, as well as things like different crystallinities and different sources. And we also have some samples which have been weathered. So we'll be comparing the effect of weathering or no weathering as pre-treatment. And additionally, each different polymer has been prepared to 2 different particle sizes. So either 100 to 500 micrometers or 500 to 2000. So again, 2 different sizes just to see if substrate size is also going to affect the rate or the sort of accessibility to biodegradation. And the experimental strategy that we're taking is set out here in this slide. And what we will do is we'll be obtaining a soil sample, which will be rich in soil microbes. And we will resuspend this in water, which will liberate the microbes which are adhered to the surface of any soil particles. You just simply leave this to stand, and once the soil particles have settled, then the top aqueous phase becomes the inoculum. And this fraction can then be used to inoculate cultures containing the array of different polymer types. So we have 6 polymer types that have been milled to 2 different sizes. We'll also be including a negative control that doesn't contain any plastics, so this should have no carbon in it. And when we take into account the replicates and also control tubes where we won't include any inoculum, then this is going to give us towards 80 tubes in total. So these cultures will all be incubated, and we will then carry out regular sampling, particularly for the first week, and also sampling at a much later point for comparison. And we'll be monitoring several different aspects of these cultures each time. We'll be looking at how well they grow. We will be plating the colonies for identification and to understand the number of colonies present. We'll be analysing the polymer, carrying out chemical analysis to look at the intermediates, and also recovering the bacterial pellet for metagenomics. So these aliquots taken at each stage of the experiment will be used in these 5 assays. So monitoring growth is quite important, and this will give us an indication of some of the growth kinetics as well. All of the samples will need to be undergo a dilution series so that they can be plated out to allow us to count the colonies and work out how many cells per milliliter we have in each culture. Bacterial pellets can initially just be stored, and by recovering these bacterial pellets for metagenomic analysis, we'll be able to look at how the population of bacteria changes at different time points during this degradation process. And by recovering the plastic particles, they can be sent to Polymateria in collaboration, and Polymateria will conduct the GPC analysis, and this will sort of show how the polymer changes over time. And finally, chemical analysis is likely to involve LCMS and NMR, and we hope that this will assist us in identifying any water-soluble intermediate products that are present in the supernatant of our samples. And by identifying the intermediates, that should give an indication as the type of genetic or enzymatic mechanisms which are at play, allowing that degradation. So in this project, then we're hoping that the outcomes will allow us to identify whether soil-dwelling organisms are likely to degrade pre-treated plastics over untreated. The reason for using soil-dwelling organisms is simply because these are the ones which are most likely to encounter fugitive plastics in the environment. And the pre-treatment process is similar to weathering effects that fugitive plastics are likely to be exposed to as they're left exposed to the elements in the environment. This initial polyethylene experiment will also help us to see whether or not the particle size has an effect on the ease or the speed of degradation. And we'll also gain an understanding into the benefits of including the Polymateria technology and as to whether that aids the speed of degradation. And finally, the chemical profiling is going to provide an indication of the intermediate products that are formed, and this will sort of inform us of the likely metabolic pathways which are responsible for the activity that we see. So once we've conducted this experiment into polyethylene, then the next steps are going to be to look into polypropylene as the other very commonly used polyolefin. And in this work, we're also going to address the use of additives in polymer production. So additives can be included in polymer production for a whole host of reasons. They include things like antioxidants, stabilizers, plasticizers, and dyes, which are included either to enhance the product performance or make the product more appealing to consumers, which is often the reasoning with using different color pigments. And these can sometimes be problematic when it comes to recycling. If you're trying to recycle a pure product, then sometimes these additives can cause issues, particularly in chemical recycling processes. An advantage of using bacteria is that you may be able to overcome some of the challenges where you have a more mixed substrate to deal with in process. So the experiments here, we can repeat the polyethylene experiment but using polypropylene. So again, looking to soil microbes as a means of biodegradation. But we're also keen to explore these additives and see how they interact when we bring bacteria into play. So we'll be testing for potential toxicity effects, as some of these, some bacteria may not be tolerant to these additives at all. But we'll consider if any of these additives may have potential benefits to the microbes. We could see from one of Murin's experiments that including a rich medium helped biofilm formation and degradation. So it might be that some of these additives, again, if they provide a benefit to the bacteria, they could actually aid or speed up the degradation process. So this is work that's still to come. So sort of to summarize the IMSE project, where we've got to so far and what we hope to achieve, in our aims and objectives, then we will hope to manufacture sustainable polyolefins which have different formations to improve biodegradation. And we will be exploiting microorganisms' biodegradation abilities and determining any intermediate products that are formed in this process. And we expect the outcomes of this will enable us to design polyolefin systems which have tuneable biodegradability. And if we then consider what the anticipated impacts are, then we hope that this ultimately is going to accelerate biodegradation of fugitive plastics that make their way into the environment and avoid refuse and recycling processes. And in doing this, we hope to reduce plastic pollution, particularly that which reaches oceans and waterways. If plastic can be broken down while it's still on the land, then it should really reduce the amount that's entering our oceans and causing the high levels of pollution that we're all well aware of. So I think that's my last slide. So I'd just like to thank by acknowledging everyone who's been involved in this work so far. So in life sciences, then Jose, who's been involved in this project, Saren, who's contributed to some of practical work and supervision of Murin, who was in the lab last year. In aeronautics, you have Kunyang, and also at Polymateria, Florence, who's carried out some of the GPC experiments, and they've also contributed in terms of consumable budget for this IMSE project. So thank you very much for listening, and I'd be happy to try and answer any questions that people may have. Thank you.
Thank you so much, Alice. That was a really fantastic presentation. And it's also really great to hear some of the fantastic work that's going on. And it sounds like a really interesting project that has a lot to bring to the world as well. So I guess one of the first questions, and I'd like to remind the audience that you can please put your questions into the Q&A function to be answered. One of my questions is, where do you kind of see this project going, or at least the research on this project going within maybe 5 years or 10 years time?
So I think identifying sort of some of the most important aspects of polymer design which are likely to lead to better biodegradation, so that we can really sort of design new polymers, particularly those that are used in plastic packaging products, which have a much greater chance of degrading more quickly. So whether that means the polymer composition, the use of different polymers, the inclusion of different additives, or perhaps certain additives or supplements that should be avoided. So really gaining a much better understanding of what it is in terms of the polymer composition that makes it more attractive to biodegradation.
Great. And so with regards to this project, what do you see being kind of the challenges that you might encounter with going forward with this project?
So I think one thing that can be quite tricky when it comes to biodegradation or enzymatic processes is that they can be a lot slower than some alternative processes. So getting the conditions quite right can be difficult. So optimizing those, particularly, it's one thing carrying out experiments in a very controlled environment, but if you're hoping to develop something that is then appropriate to be deployed outside in the real world, then it can be difficult to really control those sort of reaction conditions. So one particular advantage to Polymateria's technology is that they've taken great steps to overcoming this. So I mentioned that a lot of compostable products that we use, so most people may have had sort of plastic drinks cups that say they're compostable. They are technically, but only really under high pressure and heat in more industrial composting facilities, whereas the technology that Polymateria have developed means that degradation can happen sort of out in the environment in the UK without sort of the need to really create very sort of special conditions, particularly if those special conditions require energy or a lot of either expense or particular sensitive conditions. So I think coming up with degradation which can carry out quite robustly is a bit of a challenge.
And so how easily do you see this project kind of feeding into a sort of how do I put it, maybe a like more commercialized system or kind of real world availability for everyone?
So I think initially there's a lot of things to try, partly because within the polyolefins there are different, we've got polyethylene and polypropylene, and within that there are different formulations that can be made. So I think there's a huge amount to explore within that. So I think the initial steps will be probably casting the net quite broadly to explore a lot of different materials with different formulations. But in terms of commercialization, then it would be looking to see those which are really showing the most promise, I think, and yeah, sort of homing in on those to push those forward.
Fantastic. So I guess maybe a final question for me, and please to the audience, please feel free to put any questions into the Q&A section, is I know that you touched on it a little bit, but what are the next steps for this project? Kind of where are we going with it next?
So I think identifying really promising microbial candidates is one of the key steps. What doesn't really fall into the scope of this project, because it's got a more limited time frame, would then really to delve into the sort of metabolic mechanisms and identify exactly what's going on with those microbes. So that's likely to involve things like genome sequencing. So that will allow us to identify which genes are responsible for particular activities. And with that, we can then hopefully improve, modify or enhance different pathways through genetic engineering. So we can take strains or microbial isolates which show promise in terms of carrying out a particular role, but through sort of techniques available in synthetic biology, we can really exploit those naturally occurring capabilities and get them to work a lot better.
Fantastic. So we do have a question from the audience, and this question is there a particular reason why those microbes will actually consume the hydrocarbons?
So bacteria, to be honest, live in quite harsh environments. So they are quite good at adapting and becoming real scavengers when they need to. So the hydrocarbons can be consumed for a whole host of metabolic processes, and as long as they're able to be taken up by these organisms. So some might require particular sort of transporters in order to be able to take the hydrocarbons that are available in the environment up into the cells. Which is why, again, the Polymateria technology is particularly promising, because it encourages the degradation to really quite relatively small hydrocarbons, so that are waxes rather than plastics. And these are a lot more accessible. But ultimately, bacteria will be looking for some sort of carbon source that they can access for growth. So that's why these sort of particular substrates would be appealing.
Thank you. So I just wanted to say, as we wrap up this webinar, thank you so much to Alice for such a great presentation, and also getting through so many questions as well. It's a really exciting project, and we're really happy at IMSE to be supporting such amazing science that's going on within Imperial. So this is the first of this series, and there will be some more webinars to come. Please do keep an eye on our Eventbrite page and on our website to be updated with more of these webinars. So thank you to everyone who attended today, and thank you especially to Alice and also everyone in the team who's working on this project as well. So thank you. Have a nice day for everyone.